Clarity Clinical Solutions.
Imagine you are developing a drug for a rare disease that affects only 50 children worldwide. A traditional clinical trial would require you to enroll about 50 patients, but you would need to randomize half of them to receive a placebo, meaning 25 children would receive no treatment for a fatal condition. That is ethically impossible. But what if you could compare your treatment against an invisible control group built entirely from historical data, patients with the same disease who were treated in the past, whose medical records are available to serve as a comparator? This is the promise of synthetic control arms, and it is one of the most important innovations in modern clinical trial design. A synthetic control arm is a comparator group constructed from existing patient data rather than by enrolling patients into a separate control arm. The basic idea is simple: all patients in the trial receive the active treatment, and the sponsor uses historical data to estimate what would have happened to those patients if they had not received the treatment. The data sources for synthetic controls vary. The most reliable is individual patient data from prior clinical trials, sometimes the sponsor's own failed or completed studies. Electronic health records provide large volumes of real-world data but require careful curation. And natural history registries track how a disease progresses without treatment, which is especially valuable for rare conditions where the disease course is well understood. A traditional randomized controlled trial enrolls patients into two groups. One receives the experimental treatment, the other receives a placebo or standard of care. Patients are randomly assigned, which balances both known and unknown confounders between groups. This is the gold standard. A synthetic control arm trial is fundamentally different. All patients receive the active treatment, and the comparator is constructed from historical data. There is no concurrent randomization. This means the sponsor does not need to recruit control patients, cutting enrollment requirements by up to 50% and saving $10 to $50 million per study. But it also means the comparison is observational, not randomized, and that introduces the risk of bias from unmeasured differences between the historical patients and the trial patients. The adoption of synthetic control arms has accelerated rapidly. As of 2024, more than 40 FDA submissions have included external control data, and over 25 drugs have been approved using synthetic control arms as part of their evidence package. The number of external control submissions has tripled since 2019. The FDA has accepted historical data windows of up to 10 years for constructing control arms, and regulators have issued increasingly detailed guidance on when and how synthetic controls should be used. In rare disease, where synthetic controls are most transformative, they have enabled drug approvals for conditions where traditional placebo-controlled trials were simply impossible. There are three main sources of data for building synthetic control arms. The first is prior clinical trial data, individual patient-level data from completed studies. This is the highest quality source because it was collected under controlled conditions with standardized outcome assessments. Many sponsors use data from their own earlier trials. The second source is electronic health records from hospitals and healthcare systems. These provide large data volumes but require significant curation, missing data, variable documentation, and non-standardized outcome definitions must be addressed. The third source is natural history registries, databases that track how a disease progresses without treatment. These are essential for rare diseases where the untreated disease course is well documented. Building a valid synthetic control arm is a sophisticated statistical exercise. The core challenge is making the historical patients comparable to the trial patients. The most common approach is propensity score matching, which calculates the probability that each patient would have been assigned to the treatment group based on their baseline characteristics like age, disease severity, and prior treatments. Each treated patient is then matched with one or more historical patients who had a similar propensity score. Bayesian approaches treat the historical data as an informative prior that is updated as the trial data accumulates. The more consistent the historical data is with the trial, the more it is borrowed. And prognostic score methods stratify patients by their predicted outcome and compare within strata. Synthetic control arms offer compelling advantages. They reduce the number of patients needed by 30 to 50%. They make trials faster because only the treatment arm needs to be enrolled. They save tens of millions of dollars per study. And crucially, they are more ethical. All patients receive the active treatment, avoiding the ethical problem of assigning patients to placebo when effective treatment might exist. However, the limitations are equally important. The most fundamental is that synthetic controls cannot control for unmeasured confounders, factors that differ between historical and trial patients that were not recorded in the data. Temporal drift in standard of care can also bias results if historical patients received different background therapy than today's patients. The FDA has been increasingly open to synthetic control arms, particularly for rare diseases and oncology. In 2023, the agency issued draft guidance on using real-world data and external controls for regulatory decision-making. The guidance covers when external controls may be appropriate, generally when randomization is not feasible or ethical, and the data quality and analytical standards required. The FDA evaluates each submission on a case-by-case basis. External control data can be used at different levels of evidentiary support, exploratory, supportive, or as the primary basis for approval. The bar is highest when the synthetic control is the sole comparator, requiring strong justification, well-characterized natural history, and a large treatment effect. Synthetic control arms have been most transformative in rare diseases. When a condition affects only a few hundred patients worldwide, a traditional placebo-controlled trial is often impossible. There simply are not enough patients to randomize. And when the disease is fatal with no approved treatment, giving placebo to some patients is unethical. In these situations, well-characterized natural history data can serve as a valid control arm. The FDA has approved approximately 20 drugs for rare diseases using synthetic control arms as the primary evidence of efficacy. These include treatments for fatal childhood neurodegenerative diseases where the synthetic control came from natural history registries tracking the inexorable progression of the disease. The most celebrated example of a synthetic control arm leading to FDA approval is blinatumomab, a drug for a rare and aggressive form of acute lymphoblastic leukemia in children. The pivotal trial was a single-arm study. All patients received the drug. The control arm was constructed from over 1,200 historical patients who had received standard chemotherapy. The drug achieved a 32% complete remission rate compared to about 12% in the historical control. This was dramatic enough that the FDA granted accelerated approval in 2014. This approval was a watershed moment. It demonstrated that the FDA would accept synthetic control arms when the disease has a well-understood poor prognosis and the treatment effect is large. The measurable impact of synthetic control arms on trial efficiency is substantial. Studies show they reduce trial enrollment by 30 to 50% compared to traditional two-arm designs. Enrollment time is reduced by 40 to 60% since only the treatment arm needs to recruit. This alone can shorten the development timeline by 1 to 2 years. The cost savings are equally impressive. Avoiding the enrollment, treatment, and monitoring of hundreds of control patients saves an estimated $10 to $50 million per Phase 3 study. And crucially, in rare diseases, these efficiency gains are not just about saving money, they can make the difference between a trial being feasible versus impossible. Despite their promise, synthetic control arms face significant methodological challenges. Selection bias is the most serious. Historical patients may differ from trial patients in ways that cannot be measured or adjusted for. They might have had different healthcare access, different prior treatments, or different disease awareness. Propensity score matching can only balance variables that were measured. Temporal drift is another major challenge. If standard of care has improved since the historical data was collected, historical patients will appear to have worse outcomes than they would today, making the treatment look more effective than it really is. And data quality from non-trial sources like electronic health records is often incomplete or inconsistent. Looking ahead, several developments will expand the role of synthetic controls. AI-generated synthetic patient data could create unlimited matched controls while preserving privacy, early work shows generative models can produce patient datasets that preserve the statistical properties of real clinical trial data. Hybrid trial designs that combine a small internal randomized control arm with synthetic data augmentation may offer the best of both approaches, the credibility of randomization with the efficiency of external data. And the FDA is developing formal qualification pathways for synthetic control databases and methods, which would dramatically reduce regulatory uncertainty once a specific data source or methodology is pre-approved. So here is the bottom line. Synthetic control arms do not replace the randomized controlled trial. The RCT remains the gold standard for establishing efficacy. But they expand what we can study when randomization is impossible. For children with fatal genetic diseases, for patients with ultra-rare cancers, for any condition where a placebo arm is unethical or impractical, synthetic controls offer a path forward that simply did not exist a decade ago. They are not perfect, and their limitations must be carefully understood. But they are expanding the boundaries of what clinical research can achieve, and bringing treatments to patients who would otherwise have no trials to join.